I'd like to first thank the organizers for inviting me to be here. It's a different kind of audience than what I'm used to speaking to. You're right, it's hard to see anybody. So if you have any questions specifically at the end, take it slowly because it's probably going to be not something that I'm used to thinking about. What I'd like to do to begin is to, first of all, say that this was a very large consortium that included a number of federal agencies, and those agencies haven't kind of signed off on this presentation. I don't think it's going to be a controversial presentation. I've been involved with this project from the beginning. I was the chair of the executive committee for the first 2 years, so I'm pretty familiar with all the different characteristics, and I have a very kind of specific idea of why this experiment was so important, even independent of BPA itself. Now, the goal I'll kind of talk about did derive from the issue or the controversy around bisphenol A, but I think that this is really an important and innovative kind of collaboration that is important to think about for the future. So first of all, CLARITY is an acronym that stands for Consortium Linking Academic and Regulatory Insights on the Toxicity of BPA. It's a consortium involving scientists from a number of federal regulatory agencies, FDA, CEFSAN, the NIMH and NTP. These are all acronyms for different federal agencies within the US, but also university-based scientists that were selected through a typical grant review program. So the peer reviewers on that grant review program essentially characterize the scientific quality of the proposal, which is different from the kind of review that FDA implemented later. They were just looking for feasibility of the assay to make sure that it would fit in with the design of this large experiment. Now, the goal was to essentially perform a very large guideline GLP study that also included endpoints that perhaps are non-traditional that are being performed in different laboratories. So I'll give kind of a update about why this was performed. So first of all, there was consensus meeting in Berlin just about a year ago, not quite a year ago, where a number of people that have a very wide difference of opinion in the past came to agree on certain issues that have to do with endocrine disruptors. The first is that a chemical's potency is important, and we all know this. It's important for hazard characterization and risk, but it's not really important for hazard identification. Now, potency is kind of a tricky term because the basic science tells us that the potency of a chemical, the efficacy really, of a chemical and a hormone is going to be dependent both upon the dose or the concentration of the ligand, but also the concentration of free receptor. And what that means, at least because I'm a developmental person, what that really means is that different tissues and cells have different sensitivities to the same concentration of ligand, whether that's a chemical or a hormone. So there was also agreement that guideline endpoints don't capture the full range of endocrine disruption, and even in the case of estrogenicity, the rat uterotrophic assay, which is kind of a mainstay of the endocrine disruptor screening program, is really not predictive of the impact of chemicals, estrogenic chemicals in different parts of the body at different times during development. So this really motivated this large program. So to just kind of summarize, in the case of estrogen, estrogen controls a number of different events, both during development and in adulthood. Guideline endpoints don't fully capture those, and of course, they couldn't capture all of those. That's a very broad statement. Different endpoints will be differentially sensitive to any estrogenic compound, not just bisphenol A, but just by virtue of the biology. And therefore, hazard characterization based on a single endpoint may not be reflective of all the other endpoints. So the core study was performed at the National Center for Toxicological Research in Alabama. It was a very large guideline GLP compliant study. There was a full battery of guideline assays that were implemented there on the site. Then tissues were blinded and sent to 13 different laboratories. Now, in this case, each laboratory had a specific goal, had a specific set of endpoints or questions that they were going to answer. I want to talk later about how this design can be tweaked just a little bit for important reasons. So this modified guideline GLP compliance study was performed. The stakeholders contributed to the final design. So we had a face-to-face meeting a number of years ago where we essentially discussed all the different elements. This was a huge study, and I think that it would have been impossible for many of us to really understand what the GLP group really had to deal with. I think that one of the benefits of this was that academic scientists came to work with people doing these kind of studies on a daily basis, and we learned a lot about how each other has to work. It was a massive amount of effort for the NCTR to perform this study. So there were, in fact, 50 animals per dose group, per sex, per age in the core study. Additional animals, for example, for me, I'm a thyroid endocrinologist, and they had a separate control group for me. In many cases, they really bent over backwards to accommodate the specific needs of different investigators. Potential sources of background BPA, including diet, cages, bedding, drinking water, etc., were all evaluated by FDA. The animals were fed a diet that was free of soy and alfalfa, and that was particularly important for me as a thyroid guy because soy-based diets themselves have chemicals in it, genistein, for example, that people think of as an estrogenic compound, but also reduces thyroid function. So I can usually tell when we do thyroid hormone levels in an animal whether they were fed on soy or a casein-based diet. It's pretty obvious. So essentially what this is telling us is that the FDA really went all out to make sure this was a clean study. It was a large enough study that it's impossible not to make mistakes. Anybody who's done lab work knows that it's not always easy to control all the variables. This was a study that had many different concentrations of BPA. There's 5 different concentrations plus positive control of ethinyl estradiol, vehicle control. There was also, these were gavage daily. The pups were directly dosed as well. There was a continuous arm as well as the stop dose arm. So they had a number of features in this very large assay that could accommodate all the different investigators. And this gives kind of a review of the different kinds of endpoints that different investigators were looking at. First of all, brain transcriptomics, I think that I would rephrase that. Heather Patisol is an investigator professor at North Carolina State University, and she really looks at estrogen action in the developing brain to look at sexually dimorphic nuclei. So she was looking at evidence of estrogenicity in the developing brain. She calls this transcriptomics, but I think looking at estrogen action in the brain is a better way to think about that. Also, there's endpoints. Anna, who's here today, looked at breast development and cancer. There's prostate, was a focus by Gail Prinz. Uterine histology and gene expression by Shuk-Mei Ho. Urogenital system by Fred Vom Saal, also looking at prostate. Penile erection mechanism by Nestor at UCLA. Also, testis and epididymis by Kim Buckelheide. Ovarian function, blood glucose, adipose tissue, spleens, thyroid, brain development by me, and heart endpoints were all evaluated. So these are all kind of non-traditional endpoints, and also endpoints that are more reflective of disease and disorder and not so much acute toxicity. There have only been a few papers that have been published on this so far, so I can't give you a lot of data. There are 3 papers here. You will be able to copy these down later. 2 that I have in white are review papers. The other 3 are primary papers showing data. There's a number of investigators that are actively working on their data and going through the publication process. I think our work is finally completed, and I'm working with FDA to publish this now. But we can't really talk about all the results. Thoughts for possible future projects. I think the first thing that's important here is to recognize that this is a first ever kind of collaboration between academics and the regulatory people, and there were bound to be some bumps, and there were. So I think that one argument is that we need greater development and organization of the goals and strategies before the RFA, which is a request for applications for funding. Also, early detailed descriptions of the animals in the samplings, including statistics, et cetera. Much of this can be figured out ahead of time. Academic researchers do this routinely. Before we start any study, we've got it fully defined. It doesn't always go the way we want it to go. But in labs like mine, at least, if something goes terribly wrong, you just start over. You can't do that with an experiment that has 4,000 animals. Also, I think that it's important to think about this design a little bit differently, and I kind of want to talk about this just for a second. So instead of thinking of capturing endpoints to evaluate the toxicity of a single chemical, we could use this kind of collaboration to identify and validate endpoints that could be included later, for example, in OECD guidelines. I'm a thyroid guy, and I think about this kind of thing quite a bit. So if you had the core study that captured specific tissues that were blinded and sent out to 3 labs so that there's duplication or triplication, I think that this would be a way of validating novel endpoints that are useful and practical and could be used within a contract lab setting very quickly and cheaply. If you consider what it took to validate the tier 1 of the EDSP in the US, I think it was about 10 years and $100 million. This could be done in just a few years and nowhere near that kind of money, although I would accept it. So I also think in performing this kind of collaboration, you also distribute the blame and the responsibility. And I think that there are cases where we need to have additional endpoints to capture endocrine disruption. This just kind of outlines this kind of concept. So thank you for your attention.